Hello everyone, my name is Daniela Quintana Martinez, and today I'll be presenting a study called Sleep Stability in Isolated REM Sleep Behavior Disorder, Parkinson's Disease, and Dementia with Lewy Bodies. This study explores how these three neurological disorders affect a person's sleep, especially in the deeper stages beyond REM sleep. This research was done by Paulo Bugalho and Marta Magrico. Both authors study sleep disorders and neurodegenerative diseases, so this research fits right into their area of expertise. The central topic of this study is how sleep patterns differ between IRBD, Parkinson's disease, and dementia with Lewy bodies. These three conditions are related because they all involve alpha-synuclein buildup in the brain. The researchers wanted to know whether sleep changes could show how these disorders progress or relate to one another. This article focuses on non-REM sleep, which is less studied in these disorders. The authors looked at how stable sleep was, how often patients had arousals, and how many sleep cycles they completed during the night. They also questioned whether the three disorders might actually represent different stages of the same disease process. To answer these questions, the researchers used one-night video polysomnography, or PSG. This test records brainwaves, breathing, muscle activity, sleep stages, and body movement. They measured things like transitions between sleep stages, the stability of those stages, how often arousals happened, and how many full sleep cycles were completed. They compared the three disorders and also looked at people with and without REM sleep behavior disorder. The study included 76 participants in total. There were 54 people with Parkinson's disease, 24 people with dementia with Lewy bodies, and 21 people with isolated RBD. Every participant completed a PSG sleep study. The mix of people with and without RBD allowed the researchers to compare not just disease type but also whether having RBD made a difference. The results showed that the three groups did not differ much in overall sleep stability. However, people with Parkinson's disease had more arousals during the lighter sleep stages, N1 and N2. They also had fewer sleep cycles overall compared to the other groups. But when the researchers adjusted for medication, especially dopaminergic drugs used for Parkinson's, these differences mostly disappeared. This suggests that medication may be playing a major role in the sleep problems seen in these disorders. Here are some strengths and weaknesses of the article. A strength is that it uses objective sleep data, and it directly compares three related neurological disorders. A weakness is that there was no healthy control group, so we can't compare these patients to normal sleep patterns. Another issue is that the participants were taking different medications, which strongly affects sleep. And finally, the data came from only one night of sleep, which might not reflect a person's typical sleep behavior. To make the information easier to understand, I recommend using visuals like a simple sleep cycle diagram, a picture of the brainstem regions that control sleep, and a bar graph showing arousal levels. These visuals help the audience clearly see the key concepts being discussed in the study. Overall, the study shows these disorders share similar sleep problems, and medication may explain most of the differences. Thank you for listening.